Hello guys, welcome to our channel Niperian Solutions. So today we are going to discuss about the biochemistry session. So we are going to cover that are the major topics in the biochemistry, right? So these are the cell, carbohydrates, lipids, enzymes and vitamins. Because nucleic acid and amino acids that we have already covered in the same playlist of FDA Maharashtra, you are getting these two topics, nucleic acid and amino acids. So whatever the remaining topics, that carbohydrates, lipid, enzyme, vitamins, that we are covering in this session. Okay, so let's start. So start with the cell, that biomolecule cell topic. So here are the different molecules of cell, like nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, right? So these all are the molecules present in your cell. So nucleus, we are talking about the nucleus. So first point is your, that is the largest cellular organelle, right? What is the nucleus? That is the largest cellular organelle. The nucleus of the eukaryotic cell contain a dense body known as nucleolus, right? So what is the nucleolus? That is the dense body, right? Then Hutchinson and Guilford-Progeria syndrome, right? Result of the distortion of nuclear envelope. So whatever the membrane or whatever the envelope is there, so distortion is there. So that's why this syndrome is occurs, right? And accumulation of abnormal protein, namely a lamina A. So where that protein is present, lamina A, it is present in that nuclear envelope. Okay. So that is about the nucleus. Then endoplasmic reticulum. So as we know, the endoplasmic reticulum is the site of your like ribosomes, right? But in which one? That is the rough endoplasmic reticulum. In the smooth endoplasmic reticulum, right? So the first one, the smooth endoplasmic reticulum, it does not contain the ribosomes. So it is involved in the synthesis of lipids. Okay, but in rough endoplasmic reticulum here, it contains the ribosomes and it is the factor of your protein biosynthesis. Right? Rough endoplasmic reticulum is disrupted to form a small vesicle known as a microsomes. Okay, this is about your endoplasmic reticulum. The next Golgi apparatus is there. What is the Golgi apparatus? That eukaryotic cell contain a unique cluster of membrane vesicles known as dictyosomes, which forms the Golgi apparatus. Means your dictyosomes convert into the Golgi apparatus. So please remember this word dictyosomes. Okay. Next, it involved in the membrane synthesis, particularly for the formation of intracellular organelle, that peroxisome and lysosomes. So these are also organelle that we will see later. Okay, so this is formed from Golgi apparatus. Then, as we know, mitochondria, that is the powerhouse of cell, the powerhouse of your cell. Then apoptosis. What is apoptosis? That is the programmed cell death or the natural cell death. Okay, so apoptosis is nothing but your programmed cell death. Okay, so next one, peroxisome and lysosomes. So just we have seen, so from which organelle these two organs are formed, or these two organelle has formed. This is from the Golgi apparatus. Here only, right? We have seen, right? Peroxisome and lysosomes. Okay. So first of all, peroxisomes, these are also known as the microbodies. It contains the enzyme catalase, right? So what catalase? So catalase protect the cell from the toxic effect of H2O2, that is the hydrogen peroxide, by converting it to the water and oxygen, right? Means H2O2 is a toxic, so that toxicity is prevented by your enzyme catalase, right? Then plant contain glyoxysomes, a specialized type of peroxisomes which are involved in the glyoxylate pathway. Okay, peroxisome biogenesis disorder. So you have to remember what is the disease. That is the Zellweger syndrome, and what it is, that is absence of peroxisome. Okay, if the peroxisomes are not present, so that disease is there, Zellweger syndrome. So you have to remember this one. The next one, the lysosomes. So these are the digestive tract of the cell. Okay, then lysosomal enzyme are categorized as hydrolysis enzyme, right? Similarly, peroxisome are catalase, lysosomes are the hydrolases. Lipofuscin. Lipofuscin is the age pigment or wear and tear pigment which has been implicated in aging process. That is also very important. You have to remember the lipofuscin. Okay. Then inclusion disease or eye cell disease, that absence of certain hydrolysis in the lysosomes. So that is the inclusion cell disease. Okay, and it is also called as the suicide bags. Why it is called a suicidal bags? Because this is the digestive tract of the cell, means that is like engulf itself, right? So that's why it is a suicidal bags. Lysosomes are also known as suicidal bags. Okay, the next topic is your carbohydrate. So whatever the important points are there, I am trying to cover that all the points like in summary. Okay, so what are the carbohydrates? These are nothing but your like polyhydroxy ketone or polyhydroxy aldehyde. So these are the compounds, are nothing but your carbohydrates, whatever the sugars are there, right? So here are the examples of monosaccharides, right? So monosaccharides are triose, tetrose, pentose, hexose, heptose. These are the examples. So depending on aldose and ketose, right? So these two are differentiating aldose and ketoses, right? So glyceraldehyde, erythrose, ribose, glucose, right? So these are the aldose category. And similarly dihydroxy acetone, erythrulose, ribulose, fructose, these are the your ketose sugars, right? So reference carbohydrate is glyceraldehyde. Then glucose contain 4 asymmetric carbons and thus it has 16 isomer. So I know you guys know that formula. So what is the formula for isomers? That is the 2 raised to the n. Okay. So glucose contain how many asymmetric carbons? 4. So how many isomers are possible? 16 isomers are possible in your glucose. Then what are the enantiomers? Enantiomers are nothing but these are the superimposable mirror images. And diastereomer are the superimposable but not mirror images, right? So this point is very important. Please remember, enantiomers are the mirror images, but diastereomers, these are not mirror images of each other. Okay, then the next concept is your epimers, right? So glucose and galactose shows epimers, and glucose and mannose also shows epimers. So glucose and galactose, here glucose and galactose are the C4 position epimers, right? Mean in that C4 position they just change their positions. Okay, and glucose and mannose, these are the C2 epimers. So this is very important. Glucose and galactose, these are the C4 position, and glucose and mannose, these are the C2 position epimers. Okay, so the next one. Okay, here are some examples of your disaccharides, right? So this one is the sucrose, maltose and lactose. Okay, so these are the examples of your disaccharides, right? So what are the hydrolyzed product of the disaccharides? So sucrose, what is the hydrolyzed product? That is the glucose and fructose. Then in maltose, that is the glucose and glucose, means two glucose units are combined and form the maltose. Glucose and fructose, it forms the sucrose. And finally what is the lactose? That is the glucose and galactose. Okay, so these hydrolyzed product are very important. Then which one is the sugar is reducing and non-reducing sugar? So only sucrose is the non-reducing sugar, while these two maltose and lactose, these are the reducing sugars. Okay, so this linkage is also very important. In your sucrose there is alpha 1 2 linkage. Okay, in your sucrose alpha 1 2 linkage is there. Similarly in maltose alpha 1 4 linkage is there, and right in your lactose beta 1 4 glycosidic linkage is there. So that slide or that table is very very important from your exam point of view. So please remember, sucrose, maltose, lactose, these are the disaccharides. Sucrose is the only disaccharide which is a non reducing sugar, right? And what these are the different linkages present in the specifically disaccharides, right? I hope this slide is very clear. Okay, the next one. Here is the classification of your carbohydrates, right? Monosaccharide, oligosaccharide and polysaccharide, right? So monosaccharide, that we have seen already, right? Your glucose, that dihydroxy acetone and glyceraldehyde, these are the monosaccharide units. Okay, so just now we have seen the disaccharide also, maltose, lactose and sucrose, right? So which one is the non-reducing? Non-reducing is your sucrose. Okay, you have to remember in oligosaccharide there are 2 to 10 sugar units. Okay, this is also very important. How many sugar units in the oligosaccharide? These are the 2 to 10 sugar units. Okay, what are the example of trisaccharide? That is the raffinose, right? Then tetrasaccharide, that is the stachylose, and the pentasaccharide, that is the verbascose, right? Means disaccharide, trisaccharide, tetrasaccharide and pentasaccharide. You have to remember the name of examples, right? Then polysaccharides. What about the polysaccharides? More than 10 sugar units. Okay, again these are categorized into homo and hetero, right? Homopolysaccharide and heteropolysaccharide. So you have to remember which one is the homopolysaccharide and which one is the heteropolysaccharide. So starch, glycogen, cellulose, chitin and inulin, right? So these are your homopolysaccharide, right? And what about the heteropolysaccharides? These are the peptidoglycan, hyaluronic acid, controlling sulfate, okay, then your keratin sulfate and agar agar. So these are the example of your heteropolysaccharides. So what is here one important point? What is inulin? Inulin is nothing but the polymer of your fructosan, right? And what is the main function of inulin? Right, it measures the glomerular filtration rate, GFR. Okay, for the estimation of your GFR, inulin is used, and that is the polysaccharide of your what? That is the fructose. So fructosan is there. Okay, the next, this is the mutarotation of glucose, right? So what is the mutarotation? Mutarotation is nothing but your change in the specific optical rotation. What is mutarotation? That is the change in your specific optical rotation. That is nothing but your mutarotation. In glucose, the alpha D glucose is showing the 112.2 degree, right, and beta D glucose, it is showing the plus 18.7 degree. And what is the equilibrium mixture? That is the 52.7 degree, right? So what is the anomers? Anomers is the alpha and beta cyclic form of D glucose known as the anomers. Means alpha D glucose, that is plus 112.2 degree, that is your alpha form, right? And that beta D glucose, that is the plus 18.7, that is your beta D glucose form or beta cyclic form, right? So these two are the anomers, alpha D glucose and beta D glucose, and these are having the specific optical rotation, and that is nothing but your mutarotation of your glucose. They differ from each other in configuration only around C1, known as anomeric carbon, और उसको हम क्या बोलते हैं? हेमी एसिटाल कार्बन. So you can see guys here, right? So that is also first carbon, okay, in both alpha and beta, that is only first carbon. Means just changing the configuration of your C1, always anomers are always changing in the configuration of C1 position, right? These are not like your epimers at any carbon, okay? So always at C1 position, so anomers are always at C1 position, okay? Similarly, fructose has a specific optical rotation of minus 92 degree at equilibrium, right? Right, at equilibrium, fructose, what is the specific optical rotation of fructose at equilibrium? It is minus 92 degree. And what about glucose at equilibrium? That is the plus 52.7 degree. And please remember alpha D glucose as well as the beta D glucose mutarotation, okay? Next one, so these are the test of your carbohydrates, that is the Mollis test. So alpha naphthol is used in the Mollis test. Then Bayes test, that is for your pentose sugar, right? What is the example of your pentose sugar? That is your ribose, okay? Then reducing test for carbohydrates, these are the Benedict test, Felling's test, Barford's test. So these are used as a reducing test for your carbohydrate. So here are the different product of your, right, different product of your carbohydrate and the reducing sugar. So glucose is converted into the sorbitol, galactose in dulcetol, mannose mannitol, fructose converted into mannitol and sorbitol, and ribose is converted into ribitol. So all are in the D form only, okay? So in the last one, the oxazone test is there. So that is very well known test to all of us. So what is the oxazone formation? So these are the different crystals are formed in that oxazone test. And what is the reagent here in the oxazone test? That is the phenyl hydrazine. So please remember the name of the reagent, that is the phenyl hydrazine. Your glucose molecule react with your phenyl hydrazine and it is forming the glucose zone. So similarly all the sugars like fructose, mannose, maltose, lactose, all are reacting with your phenyl hydrazine and it is forming the respective glucose zone, okay? One important point in this one, only two carbons are involved in your oxazone test, right? C1 and C2. Please remember guys this, only C1 and C2 is involved in the oxazone test. So here also you can see, right, these only two carbons, right? So these two carbons are involved. Okay, so that's why oxazone test is important. And here are the some important points like glucose, fructose and mannose. Okay, glucose, fructose and mannose, these are showing the needle shape structure in the oxazone. And maltose, right, sorry, in maltose it is showing the sunflower like shape. And lactose is showing your powder puff shape. Here is one important point, sucrose, right? What is sucrose? That is the non reducing sugar or the invert sugar, we can call it as in invert sugar. It cannot form oxazone, right? So most of the time they are asking which one of the following is not showing the oxazone taste. So what it is? That is the sucrose, because it is a non reducing sugar. Okay, because maltose and lactose, these are also disaccharides, but these are the reducing sugars, so that's why they are showing the different forms. So maltose showing sunflower shape and the lactose are showing the powder puff shaped, but the sucrose is the non reducing sugar, so that's why it is not showing any oxazone taste. So that point is very very important. Okay, the next one, lipids. So lipids, as we know generally what are the lipids and all. So here are the examples of saturated fatty acids and unsaturated fatty acids. So from saturated fatty acids, guys, you have to remember, so just the number of carbons, right? So number of carbons present in your specific compound, especially from like valeric acid, caproic acid, capric acid, lauric acid, right? So these all are the examples of your saturated fatty acids. So you have to remember the number of carbon present in that specific acid. Okay, then from unsaturated fatty acids, you have to remember that all the things. Okay, so first of all we will see in palmitoleic acid, right? So cis 9 hexadecanoic acid, right? Then what are the another examples? These are the oleic acid, linoleic acid, linolenic acid, arachidonic acid, right? So these all are the examples of your unsaturated fatty acids. So accordingly, in this table, okay, they have giving some numbers, right? So what are those? Okay, so what is the number of 16, 18, 18, right? So first, right, this one, the first one, sorry. Okay, so this first one, 16, 16, 18, 18, 18, 20. So these are the number of carbon present in that specific molecule. What is that? First column, that is nothing but the number of carbons. Means in palmitoleic acid, 16 number of carbons are there, right? In the second column, that 1, 1, 2, 3, 4, right? So these are the number of double bond present in that specific fatty acid. Means in palmitoleic acid, one double bond is there. In oleic acid, again one double bond is there. Linoleic acid, two double bond is there. Linolenic acid, three double bonds is there. And in arachidonic acid, there are four double bonds. Please, this is very important. And what is the third one? Third one is the position of that double bond, right? Means in the palmitoleic acid, means one double bond is there, and what position? That is the ninth position. Okay, in oleic acid also ninth position is there. Then linoleic acid, that is the two double bonds are there. So what are the positions? 9 and 12. Okay, in linolenic acid, three double bonds are there. What is the position? 9, 12 and 15. And in arachidonic acid, four double bonds are there. And what are the numbers? That is 5, 8, 11 and 14. So that table from the lipids for the unsaturated fatty acids, that is very important. Okay, here are some like examples of fatty acid and their series. So means series like which omega series is there, right? So we are starting the numbering from like alpha, beta, right? So similarly the omega numbering is also there from another end side. So linolenic acid is omega 3 series. Then linoleic acid, that is the omega 6 series. Arachidonic acid, omega 6 series. And the oleic acid, that is the omega 9 series. So that table is also very very important, guys. So this whole slide you have to remember the unsaturated fatty acids. Then fatty acid with what kind of series, like omega 3, omega 6 or omega 9. And here are the number of carbons, right? So this one. So you have to remember the number of carbons in saturated fatty acids. In unsaturated fatty acids, you have to remember the number of carbons, number of double bond present in it, and at what position that double bonds are present, and the omega series, right? So I think this slide is very very important from the your lipid topic. Okay, the next one. Here are the some structures of your lipids like lecithin, cephalin, and cardiolipin. So just what is the lecithin, right? So lecithin is your phosphatidylcholine. So because we know what is the structure up to here, right? So that is the phosphatidic acid. What it is? Phosphatidic acid, and the remaining structure, this one, right? That is your choline, means phosphatidylcholine, that is nothing but your lecithin. Similarly in the second structure also, right? Phosphatidic acid combined with your ethanol amine, that is your phosphatidyl ethanola amine, that is your cephalin. And the cardiolipin, that is also very important. So cardiolipin is nothing but the, right, like that your phosphatidyl glycerol, right, means two molecules of your phosphatidic acid combining with one glycerol molecule. Okay, so that is your cardiolipin. So cardiolipin is very important. So cardiolipin is the only phosphoglyceride that possesses antigenic property, right? Here is the some spelling mistake, that is property, right? Means cardiolipin is the only phosphoglyceride that is having your antigenic property. Okay, here is the structure of like steroid. So what is the steroid nucleus? That is the cyclopentanoperhydrophenanthrin ring. So why it is cyclopentanoperhydrophenanthrin? Because the first ring, D ring, right here. So D ring is your cyclopentane ring. So that's why cyclopentano. Perhydro means there is no double bond present, right? In all the rings, all bonds are the single bonds, means perhydro. And phenanthrin, so this ring is the phenanthrin, ABC. Okay, what it is? Phenanthrin. Means D is cyclopentane, perhydro means all, means there is no double bond, and phenanthrin ring is ABC, means cyclopentano perhydrophenanthrin. Okay, so similarly here is the structure of your cholesterol, right? So please guys see the structure carefully. At third position there is a hydroxy group. Okay, here. Then again double bond present in between the C5 and C6 here. And methyl groups, how many methyl groups are there? Total 5, right? What position? Here 19, 18, 21, then 26 and 27, means total 5 methyl groups are present in your cholesterol. And finally this 8 carbon chain attaches to the C17 position. Here is the C17 position guys. Then 8 carbons are there, right? Means like 21, 20, 22, 23, 24, 25, 26 and 27. So these are the 8 carbon chain attaches to your C17 position. So these are the some important points of the structure of your cholesterol. So please remember this. Sometimes they are asking the question of the structure of the cholesterol also. Okay. Okay, the next one. Then these are the different type of lipids in our body. So these are the chylomicrons, very low density lipoprotein, then low density lipoprotein, high density lipoprotein. Okay, so among these very low density lipoprotein is a bad cholesterol. Okay, and high density lipoprotein, that is your good cholesterol, right? So if you are talking about the size of that specific like what we can say, the fatty acid or the cholesterol present in your body, so the chylomicrons having the largest size, right? Because it is almost 500 nanometer. Okay, so according to the size, chylomicron is the highest, and the uh, lowest size, that is the HDL, high density lipoprotein, means it is having the highest density but the smallest size. And chylomicron is the having the lowest density but highest size. So that is about the size and your density, right? But what about the protein content? So protein content is also similarly, like in chylomicron there is very less protein, right? In chylomicron very negligible amount of protein is there. But in VLDL, that is your 6%, again LDL, that is your 20%, and in high density lipoprotein, that is our good cholesterol, it is having the 44%. Means as size decreases, protein content increases. Okay, so the next, enzymes, right? So as we know, enzymes are the catalyst. Okay, so these are just they are increasing the rate of reactions. Okay, so enzymes are biocatalyst, the catalyst of life. Okay, a catalyst is defined as a substance that increases the velocity of rate of chemical reaction without itself undergoing any changes in the overall process. Okay, so these are nothing but the enzymes. So holoenzyme, because that is the active enzyme, right? Holoenzyme is nothing but your active enzyme, and what it is made up from? Your apoenzyme and coenzyme. So apoenzyme is a protein part and coenzyme is non-protein part. Okay, so the non-protein, organic, low molecular weight and dialysable substance associated with the enzyme function is known as coenzymes. So proenzyme or zymogens, so these are the inactive enzymes. So here is the classification. Here is the classification of enzyme, that is the oxalate, oxidoreductase, transferases, hydrolysis, lyases, isomerases and ligases. Right? So that oxalate is having your classification of your enzyme. So that is the standard classification of enzyme. Here are they are giving some numbers. EC 1.000, sorry 1.1.1.1, right? So what is this actually? What is the systematic classification? EC, what is EC? That is the enzyme commission. Okay, what is EC? That is the enzyme commission. That first one is there your class, then subclass, then subclass, and the last one your individually specific enzyme. Okay, I hope this is clear. That is the classification of enzymes. Okay, in enzymes we know there is a Michael-Menten equation, right, for your rate of reaction or what are the other aspects. So Michael-Menten constant or the Briggs and Halden's constant. So you have to remember this structure, this formula. V is equal to, right, V max into S divided by Km plus S. Here V is measured velocity, V max is the maximum velocity, S is a concentration, right? What is S? That is the substrate concentration, and Km is Michaelis-Menten constant. So here are some important points about the Michaelis-Menten constant. So we will see one by one. The Michaelis-Menten constant, the substrate concentration to produce the half maximal velocity. What is Km? The substrate concentration to produce half maximum velocity. The Km value is constant as we know, and it is a characteristics feature of a given specific enzyme, right? Means every enzyme has a specific Km value. A low Km value indicates a strong affinity between enzyme and substrate. Okay, means if Km or that constant value is low, means the stronger affinity between your enzyme and the substrate, and Km is not dependent on the concentration of your enzyme. So what is enzyme inhibition? There are the different type of enzyme inhibition like allosteric, that all this type, right? But here is the important point, like competitive and non-competitive inhibition, right? So according to the Michaelis-Menten equation or the Michaelis-Menten constant, you have to remember in Km, right, means that is the Michaelis-Menten constant. So in competitive inhibition your Km is increased but Vmax is unchanged, and in non-competitive inhibition your Km is unchanged but Vmax is decreased. So that table is very important about the competitive and non-competitive inhibition regarding your Michaelis-Menten constant. Okay. Okay, lock and key model. So who has given the lock and key model? That is the Fisher template theory, or the what is the name of that scientist? That is the Emil Fisher. Okay, then induced fit theory or Coastland model, right? So these two theory, and please remember the name of that scientist. Okay, and the third one is the unit of your enzyme activity. What it is? Kettle. Okay, so 1 kettle is equal to 6 into 10 to the power 7 international units. Okay, here is the table. So the table is very important. Please star mark this table, because most of the time in enzyme they are asking which coenzyme is derived from which vitamin. Okay, very important. So first one is the thymine pyrophosphate. So it is derived from, name itself indicate thymine, right? Then FMN, flavin mononucleotide, and the flavin adenine nucleotide, means FMN and FAD, these are derived from your riboflavin. Then nicotinamide adenine dinucleotide, NAD, means NAD and NADP, these two are from your niacin. Then lipoic acid, then vitamin is also lipoic acid. Pyridoxal phosphate from the pyridoxine. Coenzyme A, this is very important. Okay, coenzyme A from the pantothenic acid. Then tetrahydrofolate, that is the folic acid. Biocytin, that is the biotin. Then methylcobalamin and desoxyadenosylcobalamin, that is from the cobalamin. So among these Please remember this one, TPP, FMN, FAD, right, NAD, NADP, right, then coenzyme A, right. So these are the important ones, so please remember this one. This table is very very important. I am saying this is very important table, so please go through it once. Okay, here are some enzymes. The level of that enzymes are elevated in which specific disease. So amylase, right, what is the disease? That is the acute pancreatitis. Then SGPT, that is the serum glutamate pyruvate transaminase in liver diseases like hepatitis. Serum glutamate oxaloacetate transaminase SGOT, that is in heart attacks or myocardial infarction. Then alkaline phosphatase in rickets and obstructive jaundice, right. Then acid phosphatase, that is cancer of prostate gland. Then lactate dehydrogenase in heart attacks in liver diseases. Creatinine phosphokinase, that is very important in myocardial infarction. That is the early marker, right. Creatinine phosphokinase is the early marker for your myocardial infarction. Then aldolase muscular dystrophy. Then 5' nucleotidase, that is the hepatitis. And gamma glutamyl transpeptidase, that is your alcoholism. So among these, please remember this amylase is very important in your acute pancreatitis. SGOT and your creatinine phosphokinase, these two are for your myocardial infarction. But so among these two, which one is the early marker? That is the creatine phosphokinase, that is very important. And this gamma glutamyl transpeptide is also very important, right. Gamma glutamyl transpeptide is for your alcoholism. So please remember in which disease which enzyme is elevated, or these are the markers of specific diseases. Okay, so that's why this table is very important. Okay, in the next one, okay, here are the isoenzymes. So lactate dehydrogenase having the 5 isoenzymes. Creatinine kinase or creatinine phosphokinase, these are the 3 isoenzymes. Alkaline phosphatase, these are having the 6 isoenzymes. Then isocitrate dehydrogenase, these are having the 4 isoenzymes. Okay, so just similarly in creatinine kinase we have seen the 3 isoenzymes, and what are those? CPK1, CPK2 and CPK3. So in this CPK2 is very important. Why? Because it is the early marker of your myocardial infarction. So CPK1, that is the present in your brain. CPK3, these are present in your skeletal muscle. And CPK2, that is in your heart. So that's why CPK2 is the early marker of your myocardial infarction. Okay, and the vitamins, right. So this is the classification of your vitamins, like fat soluble vitamins. These are the A, D, E, K, right. And water soluble are your B complex and non B complex. So non B complex, we know the vitamin C. Again, B complex is divided in the 2 categories. Some are the energy releasing. That point is very important, right. So because we are thinking like the classification is very easy, and we know, because this point is very very important. Which one is the hematopoietic vitamins? These are the B9 and B12, and remaining are your energy releasing vitamins. So from classification you have to just remember the which are the fat soluble and which are the water soluble, and among the water soluble you have to remember which are the energy releasing and which are the hematopoietic vitamins. Okay, here are the structures of some vitamins, because what happens, sometimes they are asking the which chemical moiety present in which vitamin, right. So that's why I have put some structures over here. So in vitamin K, there are the vitamin K1, K2 and K3, right. So what is the K1? Because all this vitamin K is the derived like phyloquinone derivatives. Okay, so vitamin K1 is the phyloquinone. Vitamin K2, that is the menaquinone. And vitamin K3, these are the menadione. So please remember vitamin K1, K2, K3 and the name of that specific vitamin K. Okay, in folic acid, what is the folic acid? That is the vitamin B9. Okay, here folic acid is your vitamin B9. And what is the structure of that folic acid? It contains the pteridine plus para aminobenzoic acid, and combinedly it is called as the pteroic acid, and plus glutamate. Finally you will get your folic acid. Means pteridine, para aminobenzoic acid and glutamate, that is the structure of your folic acid. And here is the function of, like here you can see the function of your vitamin K. So what are the factors? The clotting factors, because the vitamin K, right, so it is having some specific clotting factors. These are the 2, 7, 9 and 10. So please remember this one, What are the factors involving at 2, 7, 9 and 10? Okay, in the next one, in thiamine, there are the two moieties are present, pyrimidine and thiazole. Please remember, pyrimidine and thiazole are separated by a single methylene bridge, right? So this question is also very important. Pyrimidine and thiazole are present, right, but separated by a CH2 group only. Okay, then in riboflavin there is a isoalloxazine ring, in pyridoxine there is a pyridine ring, and in biotin, that is the vitamin B7, it contains the imidazole ring plus thiophene ring. Okay, this chart is also very important because all vitamin are having their antagonist. Like vitamin K is having dicumaral and warfarin. As we know, these are the anticoagulant molecules, dicumaral and warfarin. Right, in thiamine, there are the thiaminase, pyrithiamine and oxythiamine. In riboflavin, that is the galactoflavin. Then niacin, that is the isoniazid and deoxypyridoxine. In biotin, there is avidin and des-thiobiotin. In folic acid, there is aminopterin, methotrexate and trimethoprim. In para-aminobenzoic acid, there is a sulfonyl amide. Okay, so this table is also very important because these vitamins are having their antagonist, or we can call it as anti-vitamins. And finally, that vitamin and the specific deficiency. So that is very important topic, like deficiency, which deficiency is associated with which vitamin. So we will see all the vitamins one by one, right? So this point is very important. Please make a star mark over here. Vitamin A is having night blindness, right, or we can call it as nyctopelia. Then keratomalacia, xerophthalmia and Bitot spot. So these four are very important. These all four, right? So the first four, these are associated with your like visual problems, like night blindness, keratomalacia, xerophthalmia, Bitot spot, right? Okay, and another are the retarded growth, sterility in males, urinary stones. Okay, so these are the minor one, but these first four are the regarding the visual cycle, or these are affected in due to the vitamin A. What about vitamin D? It is also called as anti-rachitic vitamin. Please remember, this one is anti-rachitic vitamin. Okay, and what are the deficiencies? Rickets, osteomalacia. Please remember, rickets in children's and osteomalacia in adults. Then alkaline phosphatase level is increased. That point is very important. Alkaline phosphatase level is increased. Among the vitamins, vitamin D is the most toxic by overdose, right? So this word is important, by overdose. Okay, right? So among the vitamins, this is the most toxic by overdose. Then what about vitamin E? Vitamin E is the least toxic among the fat soluble vitamins, right? It has contained the 6 hydroxychromine ring and antioxidant properties due to only that 6 hydroxy group. So please, these points are very important. And what is the deficiency? That is the sterility. So that's why it is also called as anti-sterility vitamin. Okay, vitamin E is also called anti-sterility vitamin. Then what about vitamin K? Just this is related to your blood clotting or the blood coagulation. And what are the important clotting factors? These are the 2, 7, 9 and 10. Okay, these are about the fat soluble vitamins. Okay, in the next one, we will see their water soluble vitamin, that is the B complex. Okay, so first one is the B1, that is the thiamine. Okay, so it is a wet berry, dry berry, right? So wet berry is associated with your serious berry characterized by the edema. And then dry berry, it is a neurological berry or the peripheral neuritis. The measurement of RBC transketolase, right? This one, what? RBC transketolase is the diagnostic test for the thiamine deficiency. Okay, so please remember the name of this enzyme, that is the transketolase. Okay, then cerebral beriberi, so that is the Wernicke-Korsakoff syndrome, right? So this is also very important. So please remember this one. Okay, what it is? Wernicke-Korsakoff syndrome. Done. Then B2 is also known as the riboflavin. Okay, it contains the 6,7 dimethyl isoalloxazine attached to the D-ribitol. So why name itself indicate riboflavin means it contains the D-ribitol and it is attached to your isoalloxazine ring, right? What is 6,7 dimethyl isoalloxazine? And what are the deficiency symptoms? Chlosis, glossitis, dermatitis. Okay, these are the deficiency. Assay of enzyme glutathione reductase. So please again remember this enzyme glutathione reductase in RBC will be useful in the assessing the deficiency of vitamin B2. Then B3, as we know, niacin or nicotinic acid, and well known disease is there, pellagra. Then dermatitis, diarrhea and dementia, that is also very important. Which disease having the 3D, right? Which disease having 3D? That is your vitamin B3. What is the 3D? These are the dermatitis, diarrhea and dementia. Sometimes it is indicated by the fourth D also, and that is nothing but your death, right? Pellagra is observed in the Hartnup disease. Okay, so please remember this disease, Hartnup disease. So pellagra is observed in your patient suffering from the Hartnup diseases. And what about B5? That is the pantothenic acid. And what is the deficiency? That is the burning feet syndrome, right? In pantothenic acid, what is the deficiency? Burning feet syndrome. Okay, then B6, also called it as the pyridoxine, right? What is convulsion, peripheral neuritis, right? So due to this deficiency, reduction in the heme production. Again here, xanthurenic acid in urine is indication for B6 deficiency, right? So please remember which acid? Xanthurenic acid. Then what about B7? That is the loss of appetite. It is also called as the biotin and also called it as anti-egg white injury factor. Okay, and in the structure we have seen it is made up of the imidazole and thiophene ring. So guys, please remember the structure of all the vitamins, at least that basic ring present in the specifically vitamin. Then what about the folic acid? These are the macrocytic anemia and neural tube defects in the fetus. And then B12, that is the pernicious anemia, and methyl malonic acid in urine is an indication of B12 deficiency, right? Again you have to remember methyl malonic acid here. Okay, and what about vitamin C? That is the scurvy, right? That is a problem related to your teeth or the gums, right, present in the teeth, right? So here we have covered in the biochemistry like that all the topics like cell and nucleic acid and amino acid that we have already covered in the same playlist in the name itself giving like nucleic acid and the amino acid. So please go through it. So I think we have covered all the important points from the biochemistry, right? So whatever the remaining, I think microbiology also remaining. So we will take one lecture on microbiology, and I think most of the important points we are covering. So just regarding your exams. Okay, so please guys keep studying and all the best for your exams. I think your hall tickets are also been released, right? So exam dates are similarly like 30 and 31 of December. So all the best for your exams. So we will see in the next lecture with any new topic or in the new subjects like compile subject, right? Similarly we have covered the organic chemistry. Now we have covered the biochemistry. One lecture we will take on any of the subject that is very important from your point of exam, right? But that is the technical one only, right? So thank you guys, thank you so much. Please keep studying, keep watching, and please subscribe the channel. Okay, thank you so much.